We will now discuss in more detail viral structure and replication. The infectious variants consist of the lipid bilayer, the envelope, where the large, middle, and small surface protein are embedded. They play an important role in the binding to target cell receptor. Core proteins for capsid, which encapsulates a genome, which is partially double-stranded DNA, and there is also reverse transcriptase labeled as polymerase. Reverse transcriptase has three activities similarly as in retroviruses: RNA-dependent DNA polymerase activity, DNA-dependent DNA polymerase activity, and RNase H activity. A majority of particles which are found in the host are non-infectious particles which only contain lipid membrane with surface proteins embedded in it. They do not contain nucleocapsid. So the question is, what is the function of such non-infectious particles? It was suggested that they may actually divert the immune responses of the host by binding antibodies and complements, so they deplete them. Therefore, the infectious DNA particle have higher chance to reach the target cells. Now, the genome, double-stranded DNA, partially double-stranded DNA, whenever it reaches a nucleus, the repair mechanism complete that gap and create full double-stranded circular DNA. Only when it is fully double-stranded, cellular RNA polymerase II can transcribe the genes. And as we mentioned, there is a surface protein right here, which encodes three surface protein. P gene encodes a polymerase, reverse transcriptase, with all three activities. C gene encodes capsid. There is also E genes, which function of which is not completely understood. Most probably it interferes with host immune responses, and X protein here in red. X protein is very interesting, and it is because it can interfere with many cellular pathways and triggers formation of hepatocellular carcinoma. Yes, X proteins is implicated in the development of hepatocellular carcinoma. So what does X protein do? It upregulates activity of tyrosine kinases such as SAR kinase. It activates RAF-MAP kinase pathway, PKC pathway, JAK-STAT signaling, NF-kappa B. All these pathways are involved in promoting cellular proliferation. In addition, it suppresses p53, it sequesters it. We know that p53 is tumor suppressor, and it is activating and stop cycling, eventually targeting cell to apoptosis if there is problems. So X protein does sequester it. It also inhibits apoptosis, which is mediated by phospholigand. So there's an enormous amount of function, which is very difficult to imagine how one protein can mediate all those, led to its name X protein, resembling X file, very secretive thing. So X protein is one protein with many different functions, which can lead to the development of hepatocellular carcinoma. Now, a virus binds to target cells via NTCP receptors, and NTCP receptor is sodium taurocolate co-transporting polypeptide. And upon binding, the viral membrane fuses with target cell membrane, and nucleocapsid is released into the host cell cytoplasm and travels into nucleus. With the nucleus, the partially double-stranded DNA is completed and form fully double-stranded circular DNA, which is normally not integrated and serves as a template on which cellular RNA polymerase II transcribes the viral mRNA. Now, under certain circumstances, hepatitis B DNA can integrate into the host genome. For many years, it was considered as some byproduct, which is non-functional. However, nowadays we know that this random integration, which is not necessary for viral replication, can actually trigger pathogenesis, contributing to the development of carcinoma, particularly if the cells is co-infected with Delta virus. Now, let's go back to replication. So this circular double-stranded DNA, which persists in nucleus, can serve as a template for synthesis of viral mRNA, which are transported to cytoplasm and give rise to proteins. Now, the viral capsid assembles and packages RNA and polymerase, reverse transcriptase. Once a nucleocapsid is formed and contains also reverse transcriptase, it starts to retrotranscription as the virus proceeds and move into ER. And the envelope is actually derived from endoplasmic reticulum membrane. Afterwards, it follows exocytic pathway through Golgi and the exocytosis out of the cells. So be aware that pre-genome RNA here is packaged by interaction with polymerase and core protein. So it is RNA which is packaged, and only afterwards reverse transcriptase start to retrotranscribe that viral RNA into DNA. Now let's compare replication cycles of retroviruses and hepadnaviruses, two families which use reverse transcriptase. However, they are very different. Retroviruses genome is single-stranded RNA. Genome of hepadnaviruses is partially double-stranded DNA. So also usage of reverse transcriptase is different. Let's look on retroviruses. When a retroviral nucleocapsid is released upon fusion into cytoplasm, reverse transcriptase retrotranscribed that RNA into double-stranded viral DNA. Afterwards, this DNA become part of the pre-integration complexes which get into the nucleus and integrate into genome. Retroviruses must integrate and form provirus in order for transcription to take place. So transcription of viral mRNA only happens from integrated provirus, and those mRNA are transported into the cytoplasm. Proteins are synthesized, virus is assembled on the membrane and released. Now, hepadnaviruses that partially double-stranded DNA with a nucleocapsid, it's entering the cytoplasm, it's transported in nucleus where the DNA synthesis is completed and forms a circular double-stranded DNA. This DNA is normally not integrated in genome and serves as a template for synthesis of mRNA. And this RNA serves as mRNA for synthesis of proteins, including capsid, but also its package, RNA is packaged into this capsid together with reverse transcriptase. And at this point, reverse transcriptase retrotranscribed RNA into partially double-stranded DNA, why only partially? Because this retrotranscription takes place as virus proceeds through exocytic pathways, and once it releases, reverse transcriptase activity, polymerase activity is done, it's inhibited, and at that point, DNA is not completed as a double-stranded, so that's why the genome is partially double-stranded DNA.